So for everybody out in virtual, you can't see the board, so you're going to have to just listen along. And maybe somebody thoughtful here who you know will take a picture. Even I could take a picture. You better do that. I know. Why not? Okay, moving on. So the part we're going to start with today is kind of where I want to finish viruses, which will take 15 or 20 minutes today, and prions, which are part of that. And then I will look at the beginning of the next unit, which none of which is on the exam when we return. That's the start of the exam 3 content. I have to figure out when and where to get that in. And that involves a chapter that's mostly about how we interact with this disease we'll introduce today, and mostly antibiotics and antivirals. So it's either 2 or like 2.5 chapters worth of information. So let us begin. This is still the Nestor chapter because, and there's in both chapters, this is the big deal about cancer. Look, depending on who you're reading, the Cowan book says whatever, 13% of all cancers are caused by viruses. Nestor, this other textbook, 15 to 30%. Suffice it to say, there's plenty of evidence to suggest that the viruses are a significant part. This is the mechanism. In your body, you have 2 mechanisms that are active that help you prevent cancers. The one that's probably the best understood, particularly vis-a-vis most of the folks in this class, are the breast cancer genes, the BRCA1 and 2, which I'm sure you have all heard about. Those are, by definition, what I've written on the board, they're what are called tumor suppressor genes. Okay? And I'll talk about those in a second. The other members of the family are called oncogenes. So those, I guess the best way to frame it is we have these 2 systems, the proto-oncogene to oncogene system and the tumor suppressor system, both play an active role in how we are constantly on the lookout for any potential cancers, changes in cells. What does that do? We'll start with the first one, which is called oncogenes. Oncogenes are the bad guys. Proto-oncogenes are there, okay, and basically your role, effectively, maybe a better way to state it, when they're inactive, they prevent tumors. Okay, we don't want them to be active. Proto means a precursor, an inactive enzyme protein mechanism. In the presence of a mutated virus, or a virus mutates your cell in a particular way, it will change proto-oncogenes to their active form, oncogenes. Those oncogenes, when activated, lead to an increased amount of tumorous growth and inevitably cancerous growth. Think of them as in your car, the accelerator. Normally it works just fine, but for whatever reason, when those mutations occur, it's like the accelerator got stuck to the floor. Your car is racing. That means your cell cycle is racing. You're making a lot of mistakes, more mistakes. Sooner or later, bad things come. Tumor suppressor genes, which are in contrast always active, are your brakes. Okay, they actively prevent, they're on sentry to prevent cancer. Viral mutation occurs, it inactivates those genes, and cancers increase dramatically. That's the real tragedy of someone who has both of those genetic markers, the BRCA2. They discover typically someone's going to write you an elective supplement. And I'd be surprised if you didn't know folks or family members. One in my family, my first cousin's wife had that. She had lost both her mom and her aunt to breast cancer. So, I mean, it's extraordinarily tragic. I don't have to tell anybody here about how tragic it is. Nevertheless, I didn't come up with the idea of the accelerator and the brakes. But I'm not that smart. There's a wonderful book. It's probably a little at the point now where maybe toward the latter portion of your college career, this author took the time to do almost like a biography of cancer through the years. and won a Pulitzer Prize. It's called The Emperor of All Maladies. You can get it on iBooks or Kindle or any platform you want to get it at. You probably pick up a used copy pretty cheaply on Amazon if you like to read, regular books like that. And it's wonderful. Very, very. And so he came up with that idea, and he explored. I mean, cancers were around, as you might guess, forever. I mean, we're talking even in Egyptian hieroglyphics, they're describing different kinds. And breast cancer, one of the earliest. Just to give you an idea. So it's a really good read. His last name is Mukherjee. I saw him do a half an hour on a TV show / radio show that I watched back. This is a good 10, 12 years ago. Very fascinating with him. Nevertheless, that's the skinny on this. So they're horrible because they have no boundaries. They go all over. They change constantly. And there's a lot of things. So the provirus can disrupt that, chronic infections and inflammation. It may have its own oncogene. This is one of the things, and you're going to see the viruses do that. But it's those two working properly that help us be the start-off point of this. For our purposes, we're not going to really look at this sort of unusual retrovirus, which is comparatively rare, that causes a particular leukemia. The big players are both hepatitis B and C, and their problem is the damage they do comes in a cell that's already replicating extraordinarily rapidly. Liver cells replicate as rapidly as any within a given organ in your body. So much so that for years when we had to do a liver transplant, it had to be somebody who died within 24 hours. Now we can do live liver transplants. A lot of liver will regenerate itself. So it's that when that virus attacks it, let's say it's a different mechanism, that those cells are already replicating so rapidly, a little damage, and certainly substantial damage, goes a long way. And it wouldn't be just hepatitis B or C. Chronic alcoholism would certainly would be similar to that. You're just because that's creating chronic inflammation. So other than the hepatitis B, and for our purposes mostly C, since in this country most everybody has been immunized for B, certainly not worldwide. B is still a big player. Is the herpes viruses, particularly Epstein-Barr, that was number 4 on your list. Herpes type 8, which is Kaposi sarcoma, which we see as an age-defining illness. Those are the big players there, and certainly and surely the leader in the field, HPV. And indeed, we look at specific subtypes of HPV when they create, whether you have received the Gardasil or whatever they're calling it today, whoever is making it, the immunization or not, that's a totally different issue. They focus, as you will see later in the course, on those certain variants that are known to be significant involvement in creating cervical cancer. So you should be familiar with this. How all this type of stuff, how they culture them, the fact they grow a lot of them on eggs, not a big deal. How viruses do most of their damage is neither one of those so much. Yes, they damage cells. They alter them. And we identify it when we stain them. They create things called inclusion bodies. And yes, sometimes they'll cause the cells to form together into what we call a syncytium. I talked briefly about RSV, respiratory syncytial virus, which is nasty. Okay? It's nasty for everybody. It's particularly nasty for people who are already sick to start. Healthy people, they still have a difficult time with it for the most part. And that causes respiratory cells to fuse that are there. That's not necessarily, as you will see in the next unit, how a lot of damage is done. A lot of it is immunological. The symptoms you get with a cold or a flu are your immune system responding. Certain viruses provoke your immune system to go absolutely crazy, for lack of a better term. It provokes such an over-the-top immune response that we can become deathly ill or die. So there's a lot. Scarlet fever would be a perfect example. That's a toxin, but it's not so much impacting you as much as it's impacting your immune system. So a lot of that's a bacterial example. Viruses, by and large, The difference between a garden variety cold and a bad flu case is the severity of the immunological response. COVID is a perfect example. We're not going to count them. We're not going to agglutinate them. And we're not talking about plants. Viroids are, again, plant-based. And I can't really, don't have any information to share with you. I do have a fair amount about prions. And there's a little bit more on this material in the Cowan chapter. I'm going to scroll through relatively quickly after this. Prions derive their name. Again, they're acellular forms. That's why they're in this unit. So they're not part of cells. They're not living things. They are misfolded proteins. Proteinaceous infectious agents. Proteins, no nucleic acid, no capsid, no envelope, no nothing. Slow, 2 to 10 years from the onset of the infection. Fatal, always. Diseases that affect humans and animals. And this is the caveat to that. And as you can see, they're always fatal. They're slow, average 2 to 10 years. Consistently, the damage is to the brain that we can see. Originally, it was seen post-mortem. And when they took out the brain to take a look at it, and they saw a brain before MRIs were commonly available. The brain was like a sponge. It was like you went out and got a sponge, and all those little holes in it that were there, hence the term spongiform encephalopathy. Okay, and those holes in the brain led to significant nervous degeneration. This was the thing that's still controversial. We don't understand them that well. We certainly don't know what destroys them, but we know they're harder than endospores. That's why they're not part of the definition of sterilization. I am not panic-stricken that I'm using something that's sterile, I'm going to magically get something like mad cow disease. I'm not worried about that because we really don't know how it's transmitted or anything like that. The unique part about that is the fact that cows that were diagnosed post-mortem with this thing that became identified as mad cow disease known as bovine cows, spongiform encephalopathy, was doing to people the same thing. Now, obviously, they're different species, so the word usually is used. And a lot of us think that this may be one agent, this prion, that with subtle tweaks affects a whole lot of different species, as you will see shortly. Kind of scary. The basic theory is this is acquired by ingestion. It's not ingesting the meat, okay? It's ingesting things that were incorporated in the meat or ground up into it. And because it affects the nervous system, that is like brain or spinal cord elements. And I'm sure none of you for lunch had brain on toast, with or without mayo, or lettuce and tomato, right? Anybody here with that? No. That's not a version of a BLT. Just saying. I thought that was clever. Not that clever. Nobody's laughing. Okay. But the reality is, here's why the cows got sick, and probably why people got sick, because, look, I hate to put it this way. When you go out and get ground meat at the grocery store, did you see them grind it? Okay, neither did I. Okay. Here's what pros do in restaurants. They grind it themselves. And they make the mixture up, and they add fat to it. And they make 80-20, they make 80-20 that way. If you didn't know it, this way it's done. No, England is, to preface, their favorite food is meat. They're very much of a meat-eating culture. You go to India, you're not going to eat meat because the cow is sacred. But chicken and lamb and things like that, fine. Great culture if you're a vegetarian or a vegan. Wonderful culture. That is there. Other areas, it's lamb intensive if you go to the Mediterranean areas. You eat what grows there or what lives there, so to speak. What they found was that they backtracked the way you do in epidemiology, and they found out where the foods that the people ate where they came from. The only choice they had was that they had to kill the whole herd. What they did years ago was when they slaughtered a cow, the meat obviously was for sale. The hide and leather, that was used for something, whatever they used parts for. Okay. The rest of it, they ground up and put it into the feed. It was called bone meal. No one knew what was in the bone meal. If there were nervous system parts, the cows ate it, it infected them, and so on. So again, it is illegal to do that. It is illegal to do that in this country. Being illegal does not mean something is not done. To this day, people in Japan will not eat meat from the US or Canada, because even though it is illegal, it does not mean they are not comfortable with the idea that it could be done. Because they're not going to get cited for murder. They're going to get some kind of a FDA ticket of some type. That kind of a thing. So the fact something is illegal doesn't mean it doesn't happen. So you have to have confidence in your source. Most of the time, I'm not worried about it. I'm as carnivorous as anybody. It's here. That's the underlying feature. So, and they used to do it for dog food, and there were a lot of dogs that got sick. In the old days, before we had really high quality dog foods, they would have things like bone meal in it. What's that ground up stuff? Sorry to disturb you, but this is, you might as well get it, to understand what it's about. So these are different diseases with different names that affect different species that are certainly similar to this. I think everybody in this list, unless I'm missing my guess, are all mammals. So obviously this can be transmitted to humans. This is how human to human transmission. They dug up mummified remains of cannibalistic tribes, did brain cuts, and found a spongiform encephalopathy. It was named Kuru. That's how disease was. I don't think the cannibalistic tribes were as unlikely to, they probably found some interesting things tasty that we would just say. You consume somebody's brains, you think you get all their intake. It's tribal. Looking at me like that could never happen. Reality is it could. This is the variant that probably hurt the people. There's a fancy pronunciation you ever hear. It's called Creutzfeldt-Jakob. There's actually an inherited type. And without going crazy over all this, a lot of us think that this can be transmitted amongst different species. The interesting thing is when proteins are irregular and affect other proteins to do so, they create tangled webs of proteins. In the brain, these are called neurofibrillary tangles. Neurofibrils are how, you remember neurotransmitters, how they move around from point A to point B. What disease has these as a hallmark? Alzheimer's. And one of my students' husbands, 15 years ago, not at this university, her husband was doing his PhD thesis at Pitt on prion-transmitted Alzheimer's. So about 5% of Alzheimer's, we are pretty comfortable saying it's transmitted by prion. So I'm not talking about something that's inconsequential, but I'm not losing any sleep over it. Okay. And over the years, I've eaten as many McDonald's hamburgers and Burger King burgers and every other burger place burger as anybody. Almost willfully. Here's what it looks like under the microscope. Not so good. And that just show how one can misfold a whole bunch. Voila. That part's done. Very briefly on any little, things that might be included in Cowan's part of the program. And I don't think there's a lot. I'm going to scroll real quickly through that. Because textbooks say the same thing all the time. So, and I don't want to go over stuff we went over necessarily that are there. We did all this stuff. And again, I like the cytopathic effects that you can detect microscopically. Changes or microscopic changes, fusion, inclusion bodies which stain, and you're able to make the diagnosis. Okay. And really, it's the accumulation, it's very well stated, of these damages that really cause us damage. A number of cells in a particular area get damaged in that area, doesn't function particularly well. This is the idea where we can see where viruses can be incorporated into our cells and alter things. Herpes certainly is a great indicator. A lot of those usual childhood diseases, they could reactivate, and people have had them. This is they call it transformation, not to be confused with that genetic movement of DNA from bacteria to bacteria. Again, oncogenic or cancer-causing viruses. And effectively, you lose controls. Here's the same group. They didn't include Hep C. They should. Herpes viruses, some. Papillomaviruses, again, some different types. Ignoring that for the time being. This is the bacteriophage thing. And this, again, if the question is about something called lysogenic conversion, typically that's where we see it a lot, is bacteria that are affected by a virus develop something that makes them more harmful, like botulism or diphtheria or cholera. Those came from genetic materials from viruses. I think there's a whole lot else to say with this. Most of the time we cultivate vaccines, we do them on bird embryos. We use eggs, duck eggs, chicken eggs. That's why what doctors typically ask for is, do you have any egg allergies if you're getting, because there's other ways they can make it. They can get one that's free of that if you're so inclined. Other things that are noteworthy, we'll look at ones as we get disease by disease, ones that are more common worldwide, ones that, and certainly these three certainly have very high mortality rates. Again, when something happens, polio, thankfully, we have not seen a reemergence of. But again, based on, as long as the rubella vaccine is of quality, when there was problems with it down the road, you'll see that in one of our presentations, what they call the rubella bulge, where we had a lot of hearing problems develop, interestingly enough. And we'll get into this as well. Okay, the relationship, if you didn't have the diabetes unit, which you probably did in AP2, big player in type 1. MS, that's another herpes virus. HHV6, we know plays a role, not sure how, different cancers we talked about, Alzheimer's, even obesity. And I think that concludes what we need there. And now that concludes all of the material that is relevant for your next exam. I'm going to take the time we have left to start to introduce this. To some extent, this ends the base chapters of microbiology, the way I do it. We now enter the realm of clinical things. We start with, you'll see the title of this particular chapter, and this is Cowan's chapter, you'll see this is week 3. We use other PowerPoint sequences and other resources as well, of course. But when I say the clinical chapters, is how it affects us, or how it affects you, or you as patients, or your patients, in whatever role you have in the healthcare area, for those going into that area. So I've covered all the worlds possible. It's fascinating. So it's going to dovetail into how things spread, epidemiologically, how we acquire them, how we release them, not always the same, how they spread, how rapidly they spread, what makes one worse than the other, what kind of damage is done, categorizing the damages, being able to understand a lot of the terminology. People go absolutely nuts when they saw the world. The minute they put pandemic with COVID, everybody went crazy. Okay, I'm not arguing that it was a pandemic. Pandemic has nothing to do with severity. You could have a common cold pandemic. Pandemic means that an epidemic has crossed effectively from a continent to another. More or less stated, crossed an ocean or a significant body of water. That's the definition of a pandemic. It has nothing to do with severity. What's an epidemic? Same thing. If we take the common cold and we should have 100,000 cases in this county in a month, and magically we had 300,000 one month, we would have an epidemic of some type of cold virus. Not going to kill anybody, but it would be of epidemic proportions. I mean, it would affect people. So when you use terms, particularly in a scientific field or a clinical field, be aware of those terms because we frighten people with terms. Sometimes it's done intentionally and I'm not saying erroneously. People thought the whole world was going to die from COVID, and they sold a whole lot. Sold's the wrong term. A lot of people made a lot of money with vaccines, and a lot of people made a lot of money with people and lost a lot of money. People not going to the workplace and jobs. Everything changed, not always for the good. So, if you're familiar with the term altruism, not everyone is altruistic. So a lot of people do things for profit, manipulate for power, for control, for things like that. So be careful when you read something, read more about something to understand what's going on with regard to these things. That's what I have to say for the time being. I'm not trying to be preachy. I'm not trying to say people did something wrong. I'm just trying to say it's wise people look at everything. So, yeah, we'll differentiate in terms. We'll look where normal microbes are, and we'll start a discussion with what they call the microbiome. If you work in a hospital with an older doctor like me, they'll talk about normal flora or normal microflora. Because we thought these were plants. They were more in the plant than the animal category. So now we call them microbes, or microbiome is all of the microbes, and how we begin to understand a little bit about that. So it's the jumping off point to understand what's going on. The first thing is what's around us or on us or in us all of the time. So the notion that people have that, oh my God, I wash my hands, everything is fine. No, you're constantly covered in microorganisms. Every square nanometer of your skin is covered in microorganisms, be they bacteria, be they viruses, be they yeast or fungi or mold. They're there all the time. Every time you inhale, you're inhaling more of them. Every single time. They're through any of your moist membranes inside, whether nasally, orally, anogenitally, doesn't matter. They're constantly there. Your whole alimentary canal, everything from your mouth all the way out, is completely covered, thank you very much, with tons, trillions of microorganisms. So let's think that don't think anything different. They play an important role. We're starting to learn about it. This was the idea of what was called the Microbiome Project. So we call them resident or resident microbes. They're on your skin. They're your buddies. They're gobbling up nutrients bad guys can be utilizing. They're covering up receptor sites bad guys can be utilizing. Okay, they're secreting products that the bad guys can't cope with. So they're there, and we have a lovely mutual relationship with them. There's a subtle change between what's called mutualism and commensalism that I ignore. Anything that wants to be on my skin or in my body or on it in some way, shape, or form, that doesn't hurt me, I'm happy with it. Mutualism is there's two species are in an area or in close contact and neither one is harmed. Effectively, they both benefit. Commensalism means one is unharmed and one benefits. Hey, as long as they're not hurting, I'm happy. Okay. I have that relationship like with spiders. If I see them on the door, as long as they don't bother me, I don't bother them. If they go in the house, no, they've got trouble. Hey, I didn't ask them to come in. Just say. So what happens, the other thing that they do is they are constantly being sampled. And inadvertently, our immune system is constantly reaching out and sampling dry barriers, moist barriers, internal structures to look to see for bad guys. And that keeps your immune system functioning. Your immune system is only helpful if you're using it. And that was, and I told the other class, and I mentioned this before, that was the misconception of what people call the hygiene hypothesis. The cleaner you are, the cleaner your area is, the less likely you are to get diseased. It's totally wrong. Okay, your immune system has to constantly sample different environments so it can adapt to and prepare to meet the challenge of potential bad guys. That's why people who live so sequestered that everything walls were washed down. This is what they did with the crown heads of Europe. Don't play with other kids. Don't go outside and play. Wash the wall down 3 times a day with lye or something else. Awful. Healthy individuals have plenty of exposure. Again, part of that went back to COVID. We needed people to be more out and for other things. We kind of lost what helped. I don't know. Yeah, I'm considerably older, more than 50 years from almost old. I mean, I don't know if your moms and dads like they threw us out of the house. Go out, don't come back for 3 hours till dinner. I think with my parents, it was like you don't have to come back at all. If you're going, have somebody call us and say, "Oh, we're here." We'll be fine. Not really. That was the idea. So, and we constantly came home filthy, dirty. I told you about measles and chickenpox parties. They wanted you to get sick. They figured it made your immune system stronger. They were right. So inevitably they get past a host of things. Most of the time it's for something dumb. You cut, scratch, scrape, something like that. That's typically how an infection gets started. You could cut yourself shaving and have that happen. Okay. So if disease is when the nature of something like that, which is about an infectious disease, really disrupts things. So a pathological disease-causing state directly by a microorganism or their product. So it doesn't have to be the organism. It can be stuff that it gives off. Toxin or something like that causes a problem. This was the idea behind the microbiome project. Okay? To understand and to sample and use really advanced techniques. Sequencers of a massive level, computer enhanced. We even do some of that stuff here with our biology students. The lab we're building over in the gym is an advanced bio lab. So we have PCR and RFLP and running a variety of units where we basically images so we can do DNA and RNA analysis and protein analysis, things like that. So we're making an investment in that. It's minor compared to what large universities have. And so we're trying to, grow with the times, so to speak. So, for instance, we saw that microbes had a lot more basically potential protein encoding genes than ours, which is interesting. There's a certain degree of complexity. I think a lot of that has to do with a lot of microbes have gotten genetic information from multiple areas. Remember, we spent that whole thing on the genetics unit talking about how sort of fragile, easily mutatable a lot of them are. They don't have our safeguards that are there. We found them in places we didn't think they were, in things that we called sterile tissues. That was really shocking. You'll see as we go along. We didn't think viruses were in our gut. Oh boy, were they ever. I mean, our gut has literally trillions of bacteria. They're vitally important for electrolytes and vitamins and things like that. And they're there because we need them in order to process foods adequately and properly. So, and we all probably have C. diff in our gut. Could be conceivably deadly. I don't think there's any question about that. I mean, it's the difference is when either they're let loose or they go to an area where they shouldn't be, or they've mutated to a strain that's particularly harmful, is when we see problems. And we've learned now that they play a critical role in our health. So we knew about all of these, all over your skin, any entry point where it gets moist. nose, throat, mouth, all the time, GI tract, openings, urethra, genitalia, vaginally, the ears, the eyes, all good. Okay. Now, in the lungs, you're constantly inhaling these things. You're not exhaling all of them. And while it takes some doing to get down into your lung, when I just inhaled, surely some got in there. When you get to the respiratory unit, they make an enormous amount, millions daily, of white blood cells that are kind of scavenger macrophages. They're like little sweepers that pick up anything that should, because your lungs interior should be sterile, but it's not completely so. That's the thing. When they don't function properly, they're clogged, they're unable to reproduce adequately. Now you're going to be predisposed to pneumonia or respiratory diseases. Again, an idiot who smoked 2 packs a day for 40 years might be predisposed to that. Certainly when they were smoking. Thankfully, less so now. I'm just saying, to me, I love the analogy it's mine. If you go to somebody's house and they like gadgets, they'll have one of those Roombas constantly sweeping their floor. You know anybody with that? Those little self-propelled things that kind of hit a wall and turn around and go elsewhere. You seen those? They sell them at Costco all the time. Must not get like that's what's in your lungs. Look where else. Bladder, okay, breast and breast milk, amniotic fluid, fetus, okay, and we've even seen it in the brain. Well, the bloodstream is not. We every time you eat, you get to some extent a transient bacteria. You get bacteria. The fact that destroyed ones still have some DNA in there is somewhat interesting. That's there. So it's different from wherever you are. Though the vast majority of people reside in a climate where we would call a temperate climate, okay, they're all over the place. Okay, the normal microbes, they're great because new ones can't really hurt them. They cover attachment sites. Okay, and like I said, waste products of theirs are useful. Remember, we derive antibiotics from microbes themselves. So when they're challenged, one of the things they make a lot of is antibiotics. That those chemicals that we call antibiotics are naturally occurring for them. Okay, and that's the whole idea between commensals and mutuals. And I'm not going to ask you how. So this was in now we seem to refer to these as comorbidities to be fancy. To me, these were people with weakened or compromised hosts, whether it was immunologically or otherwise. They could just very well have been a diabetic. They could have been someone who was older. It could be a baby. Look at all the categories. Extremes, always. I mean, we're always worried about, though, whenever there's an infectious disease, particularly diarrheal, vomiting, because fluid and electrolytes are very labile in those folks. Okay. Other types of genetic defects or age, certainly, but people who are immunologically compromised genetically, that may not be that common. And post-operatively, you're always at risk because you're devoting your resources to healing, particularly the larger the surgery. The more resources, anybody with a transplant, your immune system's already busy dealing with that. And most of the time it's dealing with it in such a way that you have to take immunosuppressant drugs. And they limit your ability. I just stopped the immunosuppressant agent I am on. Not for any reason, not what I was advised to. I'm altering my diet and my lifestyle, mostly to lose weight, but to try to take less medications. Less pill. I've given that a shot for the time being because you can always go back. And I feel I have enough overall health and motivation to do it. So when you get to immune systems, I've the last 3 years taking a drug called Humira. And Humira happens to be what it opposes something called tumor necrosis factor. Okay. And tumor necrosis factor helps you by killing tumors. I'm opposing that because of an autoimmune disorder that I have. I think I can control it better with diet and, heaven forbid, this word, exercise. Oh. going to punish my tongue for saying that word. You're bad. Any underlying diseases, agents, stressors, we talked about that with herpes infections, pregnancy's major stressor because of all the hormonal things. Again, your body can only survive. It's hard to fight multiple battles. For those of you who work on a clinical floor, you may wonder why so-and-so died or so-and-so is close to death. If they have multiple problems, your body has a tough time. Okay, you only so many battles that are in there you can fight. Some people are remarkable. I've been saying that for my mother-in-law at 97, 98, going to be 98 in August. A lot. Like the next one could be it. And she gets another RSV bout or whatever. Thankfully, hasn't happened. But you're always worried about that. Recently, uterus incontinence was to be sterile during the embryonic and fetal period. They're finding that different. That may shock you. It surprised me. Okay. Vaginally, the most abundant bacteria in the vaginal tract, which is the key protector for infection, is called lactobacillus. We learn about that in the urinary tract chapter toward the latter part of the thing. What happens, it makes, lactobacillus is very fragile, but it makes a ton of acidity. And acidity is what protects women, particularly during the childbearing years. Okay? And you can see, baby from skin disorders, other conditions, pardon me. And even human milk, 600 species of bacteria and sugars the baby can't digest, but those gut bacteria that they pick up, starting with the passage through the vaginal tract, are the big players in that. We're all born lactose intolerant. That's why milk is not a substitute for formula, okay, where breast milk is, okay, because of those factors that are in there. And people who are lactose intolerant or early on, as many people are, it takes a while. Some it takes longer to develop than others. And it works the flip way too. A lot of people who are able to use dairy all of a sudden become really bad. Two of my wife's boys are like that. I mean, oh my goodness. They could eat anything, and all of a sudden now, if they get anything dairy, they just really get sick. So, and one's gone vegan for that. She has another two of her children are vegans also. Reiterates it. And this is where I kind of got toward the end of what I was presenting in the other class. Just to give you an idea, what is it meant by being a pathogen? Disease causing. Okay. Disease causing agent is what we call a true parasite. This is the context of a parasite that's not like a fungal disease, because sometimes we call those parasitic infections, or amoebiasis or giardiasis. A parasite is any microorganism that flourishes in you while hurting you. It grows and it flourishes while you get worse. That's a true pathogen. And really, true pathogens are able to cause disease even in people with normal immune defenses. What you're going to see in the hospital, because people who are sick in the hospital are by definition almost always compromised hosts. They wouldn't be there. I'm telling you, from someone who did a lot of elective surgery, you'd much rather do it out of a hospital environment. In a surgery center or in an office a center that's designed, or an outpatient center, there's less germs, there's less sick people. The more sick people, the more germs. Just putting it in very, very base terms that are there. So what you're going to see a lot, you hear about MRSA all the time, you're going to see in the hospital-based methicillin-resistant staph infections are more often MRSA, Staphylococcus epidermidis, that, by the way, is on 100% of our skin 100% of the time. It's not methicillin resistant. You work in a hospital twice a year, you're cultivating MRSA in your nostrils, because that's where it grows. And everybody told you to keep your nose clean. Okay, just giving you. And what we do is we measure its pathogenicity in terms of what are called virulence factors. That's its nastiness. Any characteristic or structure of the microbe that makes it, its ability to establish or cause damage. How easy it is to get it. How small a dose might need to get it. How quickly it does damage. How quickly it overcomes your defenses. How quickly it can kill you. Okay, how severe are the symptoms? All of those are virulence factors. You'll see as we go disease by disease, we look at those factors. They're all important. We'll even see in this chapter some of the unique ways it gets to do that. So I think it's really important. So we'll look at going forward how much damage and how it does it and how easy you are to get it, and if you get it, can you actually get rid of it? So thanks very much. We'll see everybody on Wednesday. We will not be here on Friday. I'm sorry. I know this is upsetting you. No Friday. I will be hopefully on a plane or waiting to board a plane, going down to Jacksonville. I'll wave to you from the beach if I record a presentation.